Well, good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Blood Pressure Monitoring in Clinical Trials, a sponsor and site perspective. My name is Edward and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive and webinars work best when you're involved, so please feel free to submit questions and comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in a listen-only mode. Please note that this event will be recorded and made available for future streaming on xtalks.com. At this point, I'd like to thank BioClinica, who developed the content for today's presentation. BioClinica, an ERT company, is an integrated clinical life science solutions provider who leverages knowledge and insight to minimize risk and accelerate development in clinical trials. Through deep medical, scientific, and technology expertise, the company provides medical imaging and cardiac safety services, clinical endpoint adjudication, clinical trial software, and drug safety solutions. For more information, please visit BioClinica at www.bioclinica.com. And now it's my pleasure to introduce our speaker for today's event. Jeffrey Heilbron attended Tufts University in Boston, Massachusetts, where he completed his Bachelor of Science degree in biology with a focus on physiology. Jeff continued his studies at the American University in Washington, D.C., receiving a fellowship and completing his Master of Science in Health Promotion and Disease Management. He was employed at the American University as an adjunct professor in the Health Promotion Program. Throughout his career, Jeff has maintained his focus on the science, technology, trial design, and regulatory considerations surrounding cardiac safety within pharmaceutical development, with a special interest in hemodynamics. Jeff has presented posters and session participation at the Drug Information Association meeting, American Society of Hypertension, and at the Cardiac Safety Research Consortium. He has supported publications in cardiac safety considerations in oncology drug development and the regulatory considerations for off-target blood pressure response in compound development. And now, without further ado, it is my pleasure to hand the mic over to Jeff, who will kick off today's event. Jeff, you may begin when you are ready.
Great. Thanks so much for the introduction, Edward. Greatly appreciate it. And good morning, good afternoon, and good evening to all the attendees at this session. And again, thank you for your participation from the BioClinica and ERT research teams. This is definitely a topic that is near and dear to me and something I have a strong commitment to. And as Edward introduced with the title, our focus today is taking a look at blood pressure monitoring, specifically from the sponsor and site perspective. During this session, there will be an interactive poll activity because we do want to get your responses and share with you some insight from you, the audience. But what we're really taking a look at today, again, is that blood pressure assessment, looking at technologies as well as the application in different research environments. So what I'd like to do right now is move forward with the presentation. And this is just an overview of the discussion topics. And a lot of the focus is going to be around on what would call the safety or the off-target assessment, and taking a look at the evolution of evaluating blood pressure signals in drug development, and a brief introduction to a draft guidance that is in development. And then taking a look onwards to the different modalities and technologies for assessing blood pressure in clinical trials, and the considerations related to those designs and approach to implementing the different technologies. One of the things that I think is important is always to show how these technologies in fact have been used in actual studies. And so there'll be a case study review for that. So my goal for today is for you as participants in this meeting is to walk away with a good understanding of the technological options for assessing blood pressure, and as well as where blood pressure assessment has moved on in the clinical trial arena, and taking that perspective both from the sponsor and the site being both very critical components to collecting accurate and valuable data related to blood pressure and blood pressure change. So with that, we're going to actually go to our first poll question to get an idea of who is in the audience. And I'm going to have Edward provide the poll right now. So Edward.
All right, thanks so much, Jeff. We do indeed have our first poll question of the day, and I am launching that on everyone's screen right now to vote on. As Jeff discussed, these polls are indeed interactive, so we do encourage everyone to vote on them. This first poll question asking, please identify the organization which best represents where you presently work. And the voting options that we have offered today are, do you work in the biopharmaceutical company, in a clinical research organization, a clinical site, or a regulatory, academic, or medical institution? Again, our audience can vote today on these polls live right now. So please do choose the voting option that best describes you. Again, the first question is, please identify the organization which best represents where you presently work. Is that at a biopharmaceutical company, clinical research organization, clinical site, or are you engaged in a regulatory, academic, or medical institution currently? I'll give the audience just a few more seconds here to cast their final votes, and then we'll share the results. Okay, very good. And with that, I will close the poll and share the results. It looks like 38% of our audience voted that they work in a biopharmaceutical company. Another 38% of our audience voted that they're involved in a clinical research organization. 25% of the audience voting a regulatory, academic, or medical institution, and 0% working that they presently work at a clinical site. All right, excellent. Thank you everyone so much for voting. And we do have a back-to-back poll over here, a follow-up question, if you will. Are you aware of the draft FDA presser guidance? And this poll question, again, can be voted on live right now. The answer options here are yes or no. So again, are you aware of the draft FDA presser guidance? Yeah, and again, you're more than welcome to select one of the two options, and that's either yes, you are aware, or no. Just a few more minutes here I'll give for the final people to cast in their votes. And it looks like most of the audience has indeed voted, so thank you very much for that. I'll close this poll as well and share the results here. So 64% of the audience voting no, they are not aware of the draft FDA presser guidance, and 36% voting yes. All right, excellent. Well, thank you everyone for that. And with that, Jeff, I'll go ahead and pass the mic right back to you.
Okay, great. Thanks, Edward. And attendees, thank you for participating. I know it sounds a little bit like a game show when we're doing that, and who's going to get to go on to the next round. But it is actually helpful because then it allows us to provide you feedback during the study, not during the study, but during the follow-up on this. So getting back to the content of our discussion today, I did want to give an informal overview of the evolution of taking a look at what I'll call the off-target blood pressure response. And what we'll be discussing today is applicable to the hypertension research when we're developing hypertension drugs, but a majority is going to be focusing on that off-target, how to assess that change in blood pressure that may be associated with a therapeutic agent that is not intended to treat blood pressure. So early on in 1988, there was a very good study that you can see looking at sibutramine, which we know has an impact on blood pressure, is quite innovative in their approach in a PK sense. In 2006, a great FDA report, which really is a foundation for a lot of my understanding coming from Dr. Gelperin out of the FDA, and we'll take a closer look at that. You see a hop to 2009 in the timeline where you really start the formulation of what is going to become the infrastructure for the draft guidance, and that's looking beyond the QT interval led by two team members from the FDA, Dr. Fiorentino and Matabushi. I had the good fortune of supporting them in that discussion as well. And then the CSRC, the Cardiac Safety Research Consortium, established a think tank on this topic of the assessment of drug-induced increases in blood pressure, where we did produce a publication back in 2013. So as it continued to evolve, there were additional presentations, global in fact, but I'm going to take you over to 2015 on the timelines and the reference to the SPRINT trial. And the why I bring that into this is it has to do with the methodology of taking blood pressure, specifically office blood pressure, and what we need to do to potentially enhance the accuracy of those readings. And we'll talk a little bit about that in that section, but you'll see the reference to whether someone is attended or unattended, and whether we're using an automated device or in fact using an auscultatory device with a study coordinator or nurse in the room. In 2018, we had the release of this draft guidance, the assessment of the pressor effect, and the reference to that is at the end of this presentation. There is a bibliography of some references you can pull from. And in 2019, a very, very informative working group during the public review of the guidance that had a number of great experts present to the approach to how this guidance will look at that blood pressure change. So I had mentioned that I wanted to focus on Dr. Gelprin's the FDA report when looking at ADHD, and again referencing back that this was in 2006 and the draft guidance comes out in 2018, I wanted to highlight a couple things on each. So in the upper left corner, you'll see that we're looking at both children and adults, and the indication, of course, is ADHD. And the important component there as you go to the right is that we are seeing the fact that ADHD was a indication that was going across all age groups, so not just children, but moving into the adult population, and that in fact, the therapeutic treatment for ADHD potentially could be lifelong or a chronic treatment. And that's an important message to hold on to as we get into the background on the draft guidance. So the considerations here that she had pointed out in the report is we do need to take a look at what happens with acute versus chronic effects of drugs on, in fact, this was blood pressure and heart rate. Taking a look at the backgrounds of the patients that will be taking this additional medication, and if there are any other underlying cardiovascular disease that may in fact impact that response to the drug. So I thought this was a very important report, and I've always used it as a reference in my presentations, which takes us to now the draft pressor guidance. This is a really valuable tool, and I give accolades to the development of it because there was a lot of thought, and is a lot of thought that has gone on into this. And so this is available, of course, for discussion and review, but some of the key points here, I didn't want to go deep into the full aspect of the draft guidance, but as with many things from a regulatory perspective, we did want to take a look at the effect of a drug on blood pressure related to benefit risk. And what I want to highlight here is that it isn't so much that is someone hypertensive or not. It is a change or an increase in blood pressure, and what potential risk does that increase in blood pressure mean, and looking at the benefit risk assessment. A lot of the background on this was derived from hypertension research, and in fact that we do know that if we do see an increase of plus or anywhere from 2 to 3 millimeters in mercury, that can create an increased risk. So really the goal here is to be able to take a look at the benefit risk of a drug that may have a blood pressure increase or blood pressure signal. And again, part of the guidance was to take a look at enhancing the precision of blood pressure measurement. And as you saw with Dr. Gelprin's report, taking a look at does that indication or does that medication is the plan for short-term exposure for the individual, or is it for chronic use? That's an important consideration when we start to take a look at the benefit risk. And there is an emphasis within the guidance on using ambulatory blood pressure monitoring as a blood pressure methodology monitoring tool, because it is very well recognized as a gold standard for really taking a look at blood pressure response. And within it, there are some study design considerations. I think it is important to note that when you do take a look at this guidance, there is emphasis of looking at the intended patient population, and then if there is a potential signal, to drill down even more, that maybe you want to look at a more at-risk population, say maybe a hypertensive individual, as well as if there is a blood pressure signal. So you really start to see, again, if there is a potential risk, do we see it increasing based upon comorbid disease states? But I think the most important thing that comes through is that the FDA really does want to promote interaction in this area. So if questions do come up, I can tell you from my own experience, the interaction is very productive and collaborative on that. So I'm going to move over to the next slide. And we are going to have another poll question, simple yes or no. Some of this we may already have insight because many of you are not aware of the poll question, but did want to hand it over to Edward to ask this one.
All right, thanks so much, Jeff. And I have actually launched that poll question already for our audience to vote on. I imagine everyone knows the drill now. But just as a reminder, everyone can vote on this poll in real time. And this next question that we have for our audience here is, have you been in contact with an FDA review division or interdisciplinary review team for a blood pressure endpoint in a protocol design? And of course, as Jeff mentioned, a simple yes or no here as far as the voting options are concerned. So again, have you been in contact with an FDA review division or interdisciplinary review team for a blood pressure endpoint in a protocol design? I'll give our live audience here just a few more seconds to cast their votes, and then I'll share the results in percentages anonymously here. All right, and it looks like most of our audience is voting here. We'll give them just a few more seconds. Perfect, excellent. And everyone seems to have indeed voted, so thank you all for that. I will go ahead and close this poll, and let's have a look at the results. We have 88% of the audience voting no. They have not, in fact, been contacted with an FDA review division or interdisciplinary review team for a blood pressure endpoint in a protocol design, and then 13% of the audience there voting yes. Very interesting indeed. Thanks so much for that to everyone. And with that, Jeff, I'll hand the mic back over to you.
Great, thanks so much. And that really is valuable information because again, the guidance is just in draft right now. And part of my goal is to make sure that we share and educate directions in the industry. And so as that continues to evolve in discussions or even independent of the guidance, if questions do come up about blood pressure changes, it is very important to engage the regulators to get some guidance in that, again, a very interactive group. So in this section, we're going to take a look at blood pressure monitoring technologies and methodologies. Again, many of you may have familiarity with this, but I did want to emphasize certain things on this. And you'll see that first bullet point. And this is, if you walk away with one thing, or let's say two things, this is one I want you to walk away with. That although there are different methodologies that we're going to talk about with the office assessment, with ABPM, with home assessment, recognize that the methodologies are complementary in assessing and defining the blood pressure response. It's not one or the other, because in fact, we do know that office blood pressure is always going to be a critical component of any trial. If you take a look at it, blood pressure is always an assessment under vital signs, always in a schedule of events. So then the question is really, where is the endpoint that I'm looking at, and that you want to implement the appropriate technology to support the study design and blood pressure response. Not every study, of course, is going to have a focus on blood pressure, but you do want to ensure that you're using the right technology and the right approach when doing it. As a reference to terminology, you'll see I use automated office BP, that's an AOBP. You can now actually centrally collect that office BP using a number of different core labs have this service, as well as some of the technologies themselves. The manufacturers can provide it. ABPM that we'll talk about, really a cornerstone technology that has been used in hypertension research, so well applied and a lot of good lessons learned when we're looking at it to do the off-target blood pressure assessment. And what you're going to see is then the use of home blood pressure monitoring and remote or telemonitored home blood pressure monitoring, what we call telemonitored self-measured blood pressure. Now, why this is important? This is important because as you look at any of the guidances that are coming out, there's the recent AHA scientific position paper by Dr. Munter and team, the US Preventative Services by Dr. Chris, the ESH, the ESC, everything that we're seeing is a trend towards outside of the clinic blood pressure assessment as maybe being more accurate in the actual individual blood pressure. So with that trend, it's recognizing that the technologies and the capabilities to do that out of office assessment should be and can be integrated into a clinical trial environment. So another key point that I want you to take home, this is number 2 on the hit list, is that cuff size matters. And you say, well, that's kind of a simple concept. Well, actually, as simple as it is a critical concept. Not all cuffs are created with the same dimensions. So what one manufacturer calls an adult medium may be a very different dimension than in fact what another manufacturer does. And that is a challenge, specifically from the site level, is to ensuring that you measure the individual's arm. Arm circumference is critical in ensuring that we use and select the right cuff. From a study conduct, this is the type of information you want to have captured on a worksheet or even in a case report form, because it is an important, consistent assessment when you're using that. So it's not just by whatever the manufacturer's definition of medium is, but you want to make sure you have the actual circumference for that individual. Taking a look at auscultatory versus oscillometric. Historically, I think everyone is very familiar using whether it be a mercury or an aneroid sphygmometer and listening to the carot cough sounds with a stethoscope. And we're seeing the trend over to the automated oscillometric assessment using specific devices to do that. Now, the movement over to the oscillometric is really to reduce certain variability in the assessment, but there is still many times the place for the auscultatory assessment based upon the population and indication. So it's not to say that there's an absolute. Many of the oscillometric devices actually will allow you to use it as a manometer. The BP environment, very critical. All the guidances really focus on ensuring that the patient is situated correctly in the proper environment for accurate blood pressure assessment. We do know that environmental variability can impact somebody's blood pressure and heart rate quite quickly. When selecting a device, you do want to make sure that you've got the device that has regulatory approval and industry and scientific validations, which are available online and with the manufacturer. And something that's come up a lot more and important to consider both at the site level as well as at a study level if they're being provided centrally is the calibration, the functional calibration, the device accuracy checks. Most manufacturers provide a specification of X number of years. In addition, there's a working group at the Association of the Advancement of Medical Instrumentation that is focusing on that as well, so that there's some standardization as to what this accuracy and functional calibration check require. So moving on, I'm going to focus on now the automated office BP and the centralized automated office BP and some of the considerations. These are great discussions that are going on right now. So for example, how many BP readings should be taken for an office assessment? In the guidance, there's often reference to a triplicate reading being taken. In other documentation, you'll see that you should take 5 and take an average of the last 2. I happen to be a proponent of taking 4 and discarding the first and taking an average of the last 3. but that is a very open discussion right now as to what the appropriate sequencing of that is. Critical to that is that there is a rest period prior to that blood pressure assessment. Right now, it's predominantly at a 5-minute sit quiet before you start taking the blood pressure. And then there's actually a rest period between, usually between 1 to 2 minutes, depending upon the protocol, between each of those replicate readings. The interesting point that we started to talk about under that SPRINT trial is whether or not someone should be in the room with the patient. So should the blood pressure assessment be attended versus unattended? A really good discussion on that topic when you take a look from an article that I referenced in the back from Andreadis, who looked at attended and unattended against ABPM. And although the SPRINT trial really focused on the fact that unattended may be better, his study, although much smaller, showed that it really didn't matter as long as you used an automated office BP unit. So basically that there wasn't interaction between the patient and the study coordinator, but that they could be in the room, and they looked at that in comparison to ABPM. One thing I do often suggest in trial design is including, at least at screening, an orthostatic assessment as well, just to rule that out. Now, looking at the functionality of the device, we spoke about, we do want to ensure that there is a rest period, preferably an automated one. So again, you have a controlled situation, that you have a sequence of readings that are generated automatically, whether it be 3 or however many it may be, and the device have memory capacity so that you can have recall. Now, an area that I've found very interesting for ourselves in clinical trials is the use of the centralized automated office BP, because it allows for the digital capture of the office blood pressure and the ability to configure software that allows to control the device as to the number of readings and the sequencing, as well as to generate averaging. From a clinical trials perspective, this also provides time date stamp, and we can provide notifications back to the study site and project team as to whether or not blood pressures are over a certain specified range. And in fact, you get visual cues, as you can see on the right, related to what position the patient should be in, and then a clock that counts down. And this is provided by a number of core labs, as well as some of the manufacturers themselves. So this is the output from something like that, a very simple report. What's nice about it, again, time date stamp, the position of the individual, and then you can see on this one, if you go down to line 7, you do get an average calculation that's provided, and in this study, it included some positional assessment as well. So I'm going to then transition ourselves over into ABPM. So this is another technology. Many of you may be familiar with ambulatory blood pressure monitoring. You may not be. This is a very valuable tool, and we'll go through this. There are 2 aspects of this, specifically when we're looking at the off-target assessment, where we're trying to define a blood pressure response, as well. So we look at an early phase, might be phase 1, phase 2, with what I would call an intensive BP study. This can sometimes be correlated to what we do in the ECG world, where we're actually trying to see if we can correlate between the PK assessments, where we're seeing what is the dose concentration or dose level, and is there an associated blood pressure response. So with that, you're really trying to see what's going on, often implemented in a SAD-MAD. What's interesting about the draft guidance, and this comes up, is should we work within a healthy volunteer population or the intended patient population? The guidance does direct us more towards the work within a patient population, which does make sense, but there have been plenty of studies where we have seen this in healthy volunteers as well. It is unique into implementing that, because it may not just be implemented in a clinical pharmacology unit, it could potentially be implemented in other clinical trial arenas. I think what most people are familiar with, again, is the late phase, where we're again looking phase 2 and above. to identify that blood pressure signal. Of course, there's extensive experience using it with efficacy endpoints with a majority of hypertension drugs. But again, we're seeing it more and more and being directed to in the guidance to help define that off-target blood pressure response. And some of the case studies that I'll show will give you a good example of that. The benefit here, of course, is that we're able to get mean values of taking a look at mean daytime, mean nighttime, 24-hour averages, and being able to compare from baseline to X number of weeks on treatment, and being able to see if in fact did and was there a generated blood pressure increase to the exposure to the compound. For those that haven't seen this, I did want to include a picture of what it looks like. It is an ambulatory setting, meaning that the patient does not have to stay in the clinic. Now, again, you're going to hear me emphasize, and I wanted to make sure from a site perspective, that it is important when we look at this, that patient education is critical on how to live with it. There are do's and don'ts that need to be reviewed with the study participant. Again, we talked about the cuff and making sure we select the cuff, but things we may take for granted that do need to be taken into consideration is, patient showering, patient activities, what you can and can't do. As great as ABPM is a tool, it will have some challenges because motion artifact is disruptive to that blood pressure assessment. So it is important that the study site is familiar, as well as then being able to transfer that information over to the study participant. From a clinical trials perspective, this, as well as the office blood pressure, become important as to scheduling, as to how long is a visit going to take, how much time do I have to allocate, because now I'm focusing more on some very primary but foundational aspects of accurate blood pressure assessment. So again, when we look at the ABPM device, it's oscillometric. It's not an auscultatory device. It is generating a calculated blood pressure value. We can modify the sequence of the blood pressure as the number of readings per hour that it will take. The strength is the high number of blood pressure readings over that 24-hour period. And we can do time matched comparison to baseline and a number of different mean values off of that. So what does that data actually look like? So this is a very simple looking at a 24-hour period. I like to look at it only because it is simple, where you're looking at the time of the day on the bottom axis, and on the x-axis you're starting to see the blood pressure in millimeters of mercury. When you're looking at each of the lines, the top of the line is the systolic, the bottom is the diastolic, and then what you see are the blips underneath are the heart rate. So here on the left-hand picture, what you're seeing is a very standard look of an individual's blood pressure during the daytime hours. And then what you see here, when you get to about that midnight, that zero time point, is what we would call the dipping effect, which we would expect so someone's blood pressure to go down in the evening. That's a normal circadian rhythm, and that's the reference here is a circadian rhythm. And then you have the early morning rise, and then that return to blood pressure. That is considered a dipper. And then over here on your right-hand side, you have an individual who is a non-dipper, where you don't see that blood pressure drop. And that's an important consideration when we start to take a look at the off-target effect, as in fact, where is that blood pressure change occurring? Is it at a specific time point, or is it overall across the 24-hour period? And again, that goes back to that early phase assessment and seeing, is there a correlation to exposure to the compound, drug in the system, and is there a blood pressure response? So what does that mean from the clinical trial perspective? This is an analysis output. And when you take a look at this and you read the header, it's systolic pressure over 24 hours by visit. So this isn't just one individual. This is a whole cohort of people. And what you can see here is here's your baseline, and we'll get back to this actual study later on in the discussion. And so your baseline systolic blood pressure for this individual is here in the green. And then when you see two different exposure days to the compound, you see that in fact, the blood pressure increased. What's interesting here is that on the 2 days, a day 4 and a day 15, you don't see a dramatic change, but a constant change in that blood pressure based upon the exposure to the therapeutic treatment, which is good information to know that over X amount of time, I do not see a continued increase in blood pressure, but I do see a maintained increase in blood pressure. So now we get into some of the more key components of that protocol design. What am I looking at when I'm going to focus on this slide on the intensive focused BP study? This is that early phase where we're trying to see, is in fact there's some association with that exposure to the compound and that potential increase in blood pressure. As we spoke before, this could be done in healthy volunteers or the intend to treat. I happen to think the intend to treat is the focal point, but as part of the clinical trial process, if you do see a signal early on, it may be important to see to take a look at the healthy population and then move on to the intend to treat. And there are a couple of different ways to do that. The SAD and the MAD studies are a great opportunity to assess blood pressure, whether it be using standard automated office or using the ABPM technology. But again, very much translational science when looking to define that. And it noted that it does in the draft guidance reference taking a look at a subgroup. So if we in fact do see an increase in blood pressure in the general group, taking a closer look maybe at patients that are at greater risk like hypertension. And taking a look at the indication, whether or not the individual is going to be exposed to the drug for an extensive period of time or a short duration of time, and whether it's something such as with an oncologic indication or if we're looking at lifestyle therapeutic agents. So points to consider here. This is really where we get into, this is a busy slide, but when we take a look at this, two key things when you're thinking about ABPM. You want to talk about what my inflation sequence is going to be, how many times an hour is my ABPM unit going to go off. Do I want it to go off every 3 times an hour during the day and 2 times an hour at night? And in fact, there are a number of different protocols that look at this, and you have to take a consideration from what we've learned in clinical practice and maybe from some of the association guidelines to what we know for clinical trials and what we're trying to prove. And this was a big discussion point and still open discussion when it comes to that draft guidance as to what's appropriate as to the number of inflations, the definition of daytime and nighttime. What does that mean in that assessment? And then what we'll talk about is the criteria as to what makes a valid 24-hour period. Basically taking a look at criteria as to did the monitoring have a sufficient number of readings overall? Did it have missing data? And how will we approach that? And so you can see how the inflation sequence, the number of readings that are scheduled, as to the number of readings that are actually captured becomes a critical component because we do want consistency. We want standardization within a patient from their baseline reading to an on-treatment reading, as well as across all the patients in the study. And so this really comes to a point when you're designing your study to incorporate your statistical folks because their input is critical if we're trying to define a change in blood pressure that could be, plus 3 millimeters of mercury increase in systolic as being an important consideration. And there are a lot of resources out there, but again, the ability to engage experts as well as the regulators for further discussion on that. But I did want to show you some examples of what that criteria actually can look like. So this happens to be one that we're very familiar with and would incorporate. So criteria that we often look to incorporate into trials is that, do I have 24 hours of data? What is that minimum duration that I need for that? Then taking a look at the percent total valid readings of my scheduled versus actual. And then drilling down more into the number of valid readings per hour. Again, this seems like a very much a decision tree kind of thing. And then coming to the number of consecutive hours where I may have invalid readings in them or 0 readings. So the idea is that from my perspective, I want to limit the gaps in data because I'm going to be doing these calculations. So I don't want back-to-back hours of missing data. Now, this is again a point of open discussion that we'll take a little deeper dive into. But in addition, we also include areas like the beginning of test, because as you saw with the circadian rhythm, is there a concern with that therapeutic agent that I need to take it between a set window of time, or is that not critical with that drug? And so lots of lessons learned from hypertension. When we do that, I wish it was simpler in some cases when we're designing that, but it is an open discussion. But we do want to ensure, and why we enable or implement this type of criteria, the goal really is for consistency and standardization within a study participant as to his or her baseline and on treatment, as well as across all the participants. And that's an important message to take home, is that consistency, whether it be by technology or here by criteria. Now, this was actually brought up in the discussion by Lars Johansson from the FDA during that workshop. And again, they ran a number of simulations. He referenced is as part of his discussion, the European Society of Hypertension, which uses these criteria, which are a little bit different than what I had just shared with you. But again, looking at the percentage, total percent of valid readings, and then they, in the European Society of Hypertension, break it out by a day and nighttime number of readings that are acceptable, and they use an every 30-minute inflation across the 24-hour period. I see this as a clinical assessment. I think that in many cases, we need to be a little bit more rigorous when we're looking at hours of missed data and what would be acceptable. But again, an open discussion point. Lars went on to continue to discuss what they were looking at from the efficient ABPM criteria. And so these were the structures that, again, were initially discussed at the meeting back in 2019. And what becomes interesting here, when we go down to the number of BP readings per hour, at least 2 measurements, and there was additional analysis on sample size within the presentation as to whether is 1 sufficient, an hour, 2, or 3. I always err on the side of more in this case, and often will use 3 measurements per hour during the daytime. But as you referenced before, you'd seen in some of the criteria that there were 70%. What was interesting from my vantage point was the move to 50% valid readings, which seemed a little bit light. But there was additional analyses that they shared as to missing readings and what they had experienced, because this was all based upon the FDA's review of a number of protocols as well as a simulation. So again, you can access the presentation, and I provide direction to how to get there in the bibliography. So again, this is an actual protocol we received from a sponsor. This was the criteria that they had built in. You'll note that it looks identical to the ESH. So as at the present time, that was the model that somebody had implemented and that the FDA in their review were okay with until the guidance comes out. Again, I often think that we do miss an opportunity based upon the definition here of day and night and missed readings to really limit the risk, because I don't like to have any missed hours back to back. And in some cases here, you can have that. You can have back to back missed hours. My personal thought is you can't, because of the variability of blood pressure, you can't replace 1 hour with another. So if I am trying to compare participant A to participant B, I really want to try to ensure that there's integrity in that data. So just wanted to show that. So I am getting close on time here, so I'm going to kind of move quickly now into the home blood pressure. As I said, that's an important consideration. There's a lot going on in this area, and there can be standard home blood pressure as well as telemonitored home blood pressure. With the telemonitored home blood pressure, lots of strengths to this. It can be used really for trend analysis, and I'll take you a picture looking at that. You can also use it from a safety perspective as to setting thresholds if blood pressures go above a specific value, or in some cases below. Notification can go back to the study site as well as to the sponsor. You can also check to make sure how active the patient is for taking their blood pressure. And again, it does allow us to really manage the devices and a full audit trail. This was a presentation I had the good fortune to work with Novartis, with Fabio Becerra at the telemedicine all the way back in 2009. Fabio shared why they had implemented this for a number of good reasons. This happened to be for a hypertension trial. But what you can see with the display, this is data that would be coming into a central repository, is if you look at the bottom, you can see an extended duration. So as the ABPM was a single 24-hour monitoring, when we're implementing home blood pressure, telemonitored home blood pressure, we can really see a blood pressure change over time. And that really has a lot of value when we're looking both at trial design and when we're looking to extended exposure of a patient to a specific therapeutic agent. Now, you'll remember I emphasized the concept of the complementary use of technology. Here's your ABPM off to the left. Again, remembering that this is a 24-hour period, and I can see that change over 24 hours with a lot of readings. Again, on your right, you can then see blood pressure data captured over months as to being able to see some trend in the data. So they are complementary. It really matters what the endpoint is that you're looking to reach. You now have a number of different technologies that will allow you to do that. So let's go quickly on to this, is these two questions, and then I'll wrap it up with the BP reference studies.
All right, thank you so much, Jeff. And we will indeed try and get through these questions as quick as possible. We do have our last two set of poll questions for the day. So the first one here is a yes or no. Have you been involved with a study that included centralized or standardized blood pressure monitoring to address an off-target blood pressure signal? And again, the answer options here are yes, I have, or no, I have not been involved in a study like that. Again, our audience can answer these in real time right now and choose from the available answer options. Have you been involved with a study that included centralized or standardized blood pressure monitoring to address an off-target blood pressure signal? We'll give the audience here just a few more seconds to cast their final votes, and then we'll of course move on to the follow-up question here. All right, and it looks like most of the audience has indeed voted. Thank you so much for everyone who voted. We have 68% of our audience answering no, they have not been involved in a study that included centralized or standardized blood pressure monitoring to address an off-target blood pressure signal, and then 32% of our audience voting yes. Great, thank you all for that. And let's move over really quickly here to the follow-up question. If you answered yes to the previous question, which blood pressure monitoring technology or methodology was implemented in your study? Now, this particular poll question is a select all that apply. So you are able to select more than one answer here. And of course, we do encourage you to do so. The answer options here are, did you use ambulatory blood pressure monitoring, standardized automated office blood pressure monitoring, home blood pressure monitoring, whether it be standard or telemonitored, or did you use a other type? So again, if you answered yes to the previous question, which blood pressure monitoring technology or methodology was implemented in that study? And from these options here, you can indeed select more than one. So just remember that when you are voting. All right, we'll give the audience just a few more seconds to cast their final votes if they haven't already. And it looks like most of you have just about voted here. which is great. Thank you all so much for that. And yeah, with that, let's go ahead and have a look at those results. So Jeff, for this poll question over here, the most selected answer at 78% was ambulatory blood pressure monitoring. The second highest selected answer at 56% was standardized automated office blood pressure monitoring. And then we had a tie between other and home blood pressure monitoring, whether that be standard or telemonitored, and that was at 22%. Again, thank you all, and with that, Jeff, I'll hand the mic back over to you.
Thanks so much, Edward, and thank you participants for answering. That's really very helpful information because, again, there's a correlation. You've heard me continue to reference the complementary methodologies. And so having, where we're advancing with the automated office BP, we are doing a better job at the office level of getting accurate data, the correlation to the ABPM data, and again, the maturing of the home blood pressure data for identifying blood pressure signal and in the hypertension field and managing blood pressure. So thank you for that. So let me go into the blood pressure references studies quickly because I do want to give a chance for some questions. I specifically identified these because A, they are the off target. The indications here is rheumatology, but what these team members did was excellent, and they really went forward in designing some great studies. And let me share. So again, this is with AstraZeneca is developing the fostamatinib with rheumatology. It was a phase 2. So again, following a lot of that concept of what was going on or what is going to be presented in that guidance, where they had 2 ABPMs, a baseline and on treatment. And in fact, this team was very proactive, and they also wanted to do home blood pressure to see that trend. Now, I won't read to you the entire abstract on this, but this really highlights a question which isn't addressed in the guidance, but if I was a pharma company, I'd be real interested in this. And that's what happens if and when there is a blood pressure increase and I remove the therapeutic agent. Do I have a return to baseline? I guess that's what I'd like to know, for the action of the compound. And in fact, this group did take a look at that, and they showed that their clinic values did return to baseline levels, and that the therapeutic agent did induce elevations in the 24-hour mean, but that it did resolve after discontinuation of the therapeutic agent. That to me is extremely valuable information from a regulatory or safety perspective. This just happens to be the graphic design or graphic display. It's a little bit hard to see. I apologize for that. But what you're looking at is the baseline and the day 28 when you're looking at the red and the red triangles, and the placebo as baseline and the placebo on treatment in the blue. And so that you can see that in fact there is somewhat of an increase with exposure to the compound at day 28. That's the open red triangles. So very interesting information. So they really learned a lot about the blood pressure response to this agent, but they kicked it up a notch. And they did a follow-on study, which is, again, way forward thinking, because this is really where you want to go if you have an agent that does create a blood pressure increase. Can I manage that? And in fact, they did a study, specifically this happened to be an animal study, but they were able to correlate it back to what they had learned from their inpatient study, and that using a hypertensive agent to manage that induced blood pressure elevation. They highlight here in their summary the fact that you do have to take into consideration the individual patient, their cardiovascular health, and their capability, the homeostatic capability within the population. So it's not one size fits all, but that you can manage that increase using antihypertensive therapy. I thought that was some great work that they had done. In a similar fashion, the folks at Pfizer early on working with axitinib, another T kinase inhibitor that we do know creates a change in blood pressure, an increase in blood pressure, this group as well, using ABPM. And again, you saw this data early on when I was showing the earlier graphs. So you can see the baseline in the purple for the systolic and the diastolic, and then that increase in blood pressure. What's interesting to see here is that, again, when you go from day 4 to day 14, the red, the green and the gray, you do not see a continued increase in blood pressure, and that's critical information. So as I expose an individual to the therapeutic agent, I do not see a continued increase in blood pressure. That really gives me an understanding of that. And again, what important to remember, benefit risk. So even if you have an increase in blood pressure, depending upon the indication and the population, that doesn't mean that the drug would stop in development. You just want to define that blood pressure response. And from part of their conclusion in this study, what was interesting to see here was that the observed blood pressure response appeared to be independent of the axitinib plasma exposure. So really an open question there. I, of course, would be interested to see what happens to the blood pressure when I remove the therapeutic agent. So moving on to another therapeutic area, women's health. Had an opportunity to work with Robert Jordan and the team here as we looked at that blood pressure response. He did a great presentation on their concept and design that can be shared as well. But in this specific area, what I wanted to highlight was the conclusion, where he showed that the ABPM was a useful methodology to detect a small transient increase in ABPM accompanied by a reduction in heart rate. It did result that this trial led to an appropriate in-clinic BP monitoring during the larger clinical trial development. And again, that really is, when you read the guidance, that's really the concept. Help me understand that blood pressure change and what should I do going forward in my later phase trials. For the folks at Forest, when they were developing milnacipran for fibromyalgia, again, using ABPM in this, and again, the drug moved forward. What's important here is that in the label, it does identify the fact that there is a potential blood pressure increase and that you do need to monitor the patient's blood pressure before and then on treatment. So I think that's kind of an important wraparound, that yes, there is a blood pressure change, but as long as that there's an awareness and there's management, that you can proceed with that. So I did want to share the references. Again, this will be available. I've got even more, but just a quick summary. And with that, I know I've been talking kind of quick. I did want to allow for a few minutes for question and answers, and I will hand that back over to Edward.
All right, excellent, Jeff. Thank you so much. We will try and get through as many audience questions as possible with the little time that we have. But of course, first off, thank you for that insightful presentation. And at this point, I would indeed like to invite our audience to continue sending in their questions or comments right now using the questions window for the Q&A portion of today's webinar. So as I mentioned, I've already received some questions here, so why don't we go ahead and get started with those. Jeff, this first audience member asking, are there specific considerations for implementing and measuring blood pressure in studies within the pediatric population?
Oh, it's a great question, and an important question, because there's a lot of work going on in pediatric research. And yes, there are important considerations, because in fact, you do want to ensure that the device that you select has been validated within the population. As we've talked about, you do have the right cuff size for use in the population, as well as trial design as to whether you're using ABPM or automated office BP. Compliance for ABPM is an important consideration. And in fact, when you get to the other aspect, which is the analysis of the data, criteria are different when we look at the pediatric and up to the adolescent age than when we're looking at the adult population. So a number of considerations there, but a very critical area. There's been some really interesting work done in pediatric oncology related to blood pressure assessment, as well as you saw with Dr. Gelperin back in 2006, the discussion around therapeutic agents in the area of ADHD and other childhood indications.
Excellent. Thank you so much for that answer, Jeff. Another question for you here. Have you had any experience with orthostatic hypotension measurement?
That's a really another one that I think is important, and it can be kind of confusing if you look at the literature, because depending upon what resource you go to, it can be done from supine being lying down to standing. It can be done supine to seated to standing, or it could be just be seated to standing. And then the question is how many readings do I take in a standing position? Do I take one? Do I take two? One at 1 minute, one at 3 minutes? So it's a little bit unclear, but the key here is to standardize whatever your approach is, that you standardize it. And again, using the automated Office BP, and specifically when you do have a software program like with the centralized automated Office BP, you can really control that, giving visual cues to the site staff as to the positioning of the patient. You do have to take into consideration the population. If you're working in the area where you know you may have a neurogenic response, that you do have to be careful for patient safety as you move them from a supine or seated to a standing position. So you do need to have awareness of your population and the potential change in blood pressure.
Wonderful, wonderful. Thank you very much, Jeff, for sharing that experience with us. Greatly appreciated. Another question for you. This viewer asking, can you discuss how other hemodynamic measures like arterial compliance and pulse waveform assessment can be implemented in clinical trials?
Yeah, that's a really important question. Didn't have a chance to really discuss it on this webinar. We've have discussed it before, but right now everything we've spoken about has been about the arterial brachial blood pressure assessment, so peripheral blood pressure. So there are definitely a lot of interest in taking a look at non-invasive central pressure and arterial compliance and pulse wave assessment, because there were studies done early on, one being the CAFE study, which in fact showed that in some cases for a hypertension drug where you didn't observe a change in that brachial blood pressure, however, using centralized pressure and using, again, the central pressure at the heart, as well as some other parts of the arterial tree, that in fact you did see a change in blood pressure. So there is a lot of interest in this area. There's actually a meeting coming up, the North American Artery meeting in May, which takes a closer look at these additional hemodynamic measures. It is a little bit different than what we're familiar with, but really shows some promise when we're starting to take a look at some agents, and especially those agents that have effect at the endothelial level.
Excellent. Thanks for that answer, Jeff. Another question for you here. Which division of the FDA should we speak to if we have a question about our protocol design to address a potential blood pressure signal in a developing compound?
That's, again, it's an important message, and I think the guidance really emphasized the need to reach out. The regulators would like to engage. So the avenue to do that is your review division is where you would start, whatever therapeutic area, and then request a consult from that IRT group within the FDA. Now you can't quite mandate it, but the idea is that the IRT, that interdisciplinary review team, is really structured to be able to support all the other divisions as to approaching cardiac endpoints. So they, of course, initiated themselves specifically when we got into the thorough QT and QT assessment, but again have taken on activities, as you saw, with blood pressure and blood pressure design considerations through this draft guidance.
Excellent. Thank you for clarifying that. And thank you again for spending this extra time to answer these questions. I think we have one more here, so we'll probably call it after this one. But this viewer asking, if I am conducting a study which includes ABPM, can the sites use ABPM units or systems that they have in their clinical practice? Or sorry, in their clinic practice, should I say?
Sure. That comes up quite often. What we would normally suggest, because you're trying to standardize things, and we've had some discussions with the regulators on this, is that it's important that throughout the study that we actually use the same device and model, when doing this versus having different devices and models. So is it possible? Yes. Then it really depends upon the phase of the trial. If we're looking at a study being done in a clin farm unit at a phase 1 environment, or if we're looking at a late phase trial. But traditionally, in doing clinical trials, we do suggest that all the study sites use standardized equipment, whether it be provided by the sponsor or by a core lab, so that you enhance the standardization and quality of the technology being provided for the study.
All right, great. Thank you very much for that answer. And as a matter of fact, for all those answers, Jeff, we have reached the end of the question and answer portion for today's webinar. If we couldn't attend to your questions today, the team at BioClinica will follow up with you. Additionally, if you have further questions, please direct them to the email address showing on your screen. I've put that there preemptively here. That's jeff.heilbron@bioclinica.com. Again, thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for today's event. A survey window will also be popping up on your screen, and your participation is appreciated on that survey, as it will help us to improve our webinars. Lastly, I've sent everybody a link in their chat box. You'll be able to view the recording of today's webinar via that link, and you can also share this link with your colleagues if you'd like them to watch as well after they've registered for the recording. So I do encourage you all to do that. Once more, please join us in thanking our speaker for his time today, Jeffrey Heilbron. Jeff, thank you so much again for a very informative webinar.
Sure. Thanks, Edward. Appreciate your support and coordination, and thanks to my colleagues at BioClinica ERT for their support, but specifically for the people who are still online, the participants, because again, we really want to share that insight with you and appreciate your feedback and participation in this webinar. So thanks very much.
Indeed, indeed. Thank you. We hope you all found this webinar informative. Have a great day, everyone.